Hello, everyone. My name is Magdalena. Today, it is my great pleasure to talk to you about deep learning-aided drug discovery. Before we start, I would like to tell a few words about the plan of today's presentation. So at the beginning, I would like to discuss some things connected with drug discovery. What is it? Why are we interested in it? And next, I will tell briefly about deep learning and graph neural networks. Then I will tell more about tools that are used in deep learning-aided drug discovery. I will present a demonstration, some implementations, and then I will tell briefly about our model that aims to predict molecular properties. Finally, I will conclude my talk. So before we begin, I'd like to clarify the process of drug discovery. So here you can see a target, and this is the object in blue. And in general, let's say that this target is associated with the disease, and in real life, this target is often a protein. And now, a chemical compound, which you can see in green, is a molecule that can cure the disease or at least can help in the treatment process. However, this molecule may have undesirable properties. And in general, we would like to optimize some properties or know that the given molecule cannot be a drug because of the properties can be bad for people's health. For instance, the molecule may be toxic. And finally, when we know that the given molecule is okay in general, it can be called a drug. And in general, drug discovery is a complicated and systematic project spanned over from 10 to 15 years, which is a long journey for a drug from invention to market in practice. Meanwhile, due to the complexity of biological systems and a large number of experiments, drug discovery is prone to failure and is inherently expensive. To address these issues, of course, many researchers proposed various computer-aided drug discovery methods for small molecules and to help in this process. And there are many different stages of drug discovery. The early stage is preclinical research, and then we have hit identification and selection, hit to lead, optimization, clinical, and we go to the clinical candidates. And despite the success in existing drug discovery, traditional methods are mostly based on molecular simulations techniques, which suffer from the huge computation costs and time-consuming procedures. And of course, that limits its application in pharmaceutical industry. And yeah, as I said, the chemical space is very large. We can say that the number of molecules is like 10 to the power of 60. Yeah, and one of the methods that is commonly used is called virtual screening. Virtual screening is a computational technique used to predict potentially bioactive chemical compounds, which I will call ligands here. And these compounds bind to a specific target, which I showed at the beginning. And as I said, it is usually a protein receptor, or it can also be enzyme. And virtual screening can substantially speed up the drug discovery process to reduce its significant costs by reducing the time invested in expensive experimental assays or experimental verification. And as you can see, it consists of narrowing down an initial large library of compounds to a considerable smaller final set of hit compounds, which are then tested experimentally in vitro to confirm their biological activity. In broad terms, virtual screening approaches can be categorized into two classes: ligand-based screens and structure-based screens. And a ligand-based screening starts from usually two-dimensional or three-dimensional structure of one or more already known ligands, and then searches a chemical library for examples that in either a 2D or a 3D sense. And in contrast, structure-based screening doesn't require a priori knowledge of any ligands that bind to the target protein, but instead it involves sequentially docking each member of the chemical library against the 3D structure of the target protein and using a scoring function, so-called scoring function, to evaluate the, let's say, quality of each modeled protein ligand complex. And the scoring function is intuitively meant to serve as, let's say, proxy for the expected strength of a given protein ligand complex. And in general, the scoring function is typically built upon either a physics-based force field or an empirical function, or just a set of knowledge-based terms. And yeah, and undoubtedly, a vast number of published models are based on traditional molecular representations, which are used as the input to the machine learning model, traditional machine learning models. And here you can see an example of the output of such algorithms. Depending on the task, we can say that we work on a regression or classification problem, and of course, to hand-crafted representations. Yes. And of course, a vast number of published models are based on these representations, as I said. But first of all, there is no clear best traditional representation of chemical compounds for machine learning algorithms, and certain representations may be better for specific tasks. Nevertheless, existing traditional molecular representations fall into mainly two categories: numerical molecular descriptors and Smiles strings. And also we have a representation called a graph approach, graph-based approach, because as you can see, a molecule can be intuitively seen as a graph. Yes. So over the last few years, deep learning methods have made significant progress on numerous machine learning tasks, leading to a substantial improvement of the state of the art in many areas of research. They have also revolutionized the way problems in cheminformatics are being solved. And compared with the conventional molecular descriptor calculation, as you can see here, a deep learning-based approach performs so-called future learning, known as representation learning. And the main idea is to learn compact and expressive representation. Yeah. And of course, a few examples of current work on representation learning and its application to chemistry includes such approaches as convolutional neural networks on graphs, wave graph attention networks, graph neural networks with convolutional ARMA filters, and so on. However, satisfactory performance is generally associated with a vast amount of high-quality data available. And in practice, the lack of labeled data in chemistry that is caused by many factors that include expensive experiments and errors impedes the exploitation of the full potential of representation learning. And another vital aspect is the limited structural information incorporated into current deep models. As you may see, a molecule is not only a set of atoms and bonds, since it also consists of various molecular dependencies that are often mixed. And however, a chemical compound, as I showed previously, can be naturally seen as a graph with nodes corresponding to atoms and edges that correspond to bonds. And of course, one may learn on a molecular structure. Viewing molecular structure as graph data leads to so-called graph neural networks-based architectures, GNNs in short. And molecular graph enables to capture the spatial connectivity of different atoms and preserve molecular bonds, which, amongst others, bring real benefits. In consequence, a large number of graph-based neural networks and models have been investigated, such as passing neural networks, convolutional networks, or graph attention networks. And obviously, they were successfully molecular representation task. One advantage of graph representation is that it carries more structural information, and besides, molecular graphs as well as the subgraphs could be directly mapped to chemical structure and thus are highly interpretable, which is very important. And of course, we have quite a lot of tools that are designed to facilitate the process of drug discovery, especially for deep learning. The examples include RDKit. RDKit is just a collection of cheminformatics and machine learning software that is written in C++ and Python. Another interesting tool is a DeepChem. This is a Python deep learning library for molecular machine learning. And last but not least, we have DDL library. And DDL library is a high performance and scalable Python package for deep learning and graphs. DDL is a framework agnostic. So what I mean is a deep.
Magdalena, it sounds like your mic's maybe a little close. It sounds a little ruffled.
Sorry?
Oh, much better. Yeah, for a second, it sounded like the sound was a little muffled, but I think you're better.
Okay.
Yep, very good.
So DDL is a framework agnostic. If a deep graph model is a component of an end-to-end application, the rest of the logics can be implemented in any major framework, such as PyTorch, Apache, and so on. Yes, and of course, we have also numerous publicly available libraries. They include CAML or PubChem. Yes, and here I would like to show you a few lines of code that can be used to use a model, one of the models that is available in this framework. And the model is called GET. And here you can see an example how can we use it to create, to use the data. In this case, we want to work on the data connected with toxicity. Yes, and then we can calculate the specified property for the given molecule. So in general, this is quite easy to use it if you are able to write a few lines of code in Python. You can easily use the available functions to work on molecular data sets. And yes, finally, I would like to tell a few words about my recent work. So in my work, which is a result of my collaboration with the University of Vienna, we proposed a neural network-based architecture that overcomes the aforementioned difficulties. So our architecture comprises of two separate blocks, and either block aims to learn an expressive representation from a given compound. The first block models a molecule as an undirected graph, and the idea is to treat a molecule as a graph with nodes corresponding to atoms and edges corresponding to bonds, and of course to learn a molecular representation. The core part of this architecture is a stack of attention layers, and followed by a fully connected layer applied to form features from molecular graphs. And the second block converts a molecule into molecular fingerprints. Then a deep learning algorithm is adapted on the feature vectors, and the output is returned. And as you can see here, the two final feature vectors are concatenated here and here, and we get this representation. And the final layer is actually a regression or classification layer, depending on the needs, and it is used to estimate the output as the property value. And as it was mentioned, our model employs two stacks of graph attention layers at the first block. The first stack aims at providing atom embedding, while the second computes a chemical compound embedding. And here, in this approach, a collection of atomic and bond features are proposed to capture the detailed structure, property, relationship information. And maybe I will tell more about the experiments we have conducted. So we evaluated the performance of our approach on numerous data sets from MoleculeNet. Here, MoleculeNet is a publicly available data set. And in general, here we randomly split data set into training, validation, and test set. And furthermore, when we work on regression tasks, we use root mean squared error as the performance metric. And as mentioned previously, in order to encode a graph, one has to provide node features. And we call this step feature extraction. And in this work, we apply 12 types of features that cover both atomic and bond features. All features are one-hot encodings, except for formal charge, radical electrons, and atomic mass, which are real numbers scaled to be on the same order of magnitude. And a glance at results on the test set reveals our method as the model with the best performance for all three data sets. Also, analysis of prediction quality indicate that the maximum difference between the experimental value and the predicted one for a given compound is not large. Moreover, most of the structures are different, and it proves that our method can capture the molecular information and doesn't depend on the type of the molecular structure. Yes. And to sum up, we introduced a method that is based on a deep neural network approach. And our method can be used to predict the biological and physical chemical properties of small molecules. And our well-designed architecture employs two separate blocks of operations to learn the molecule's representation. And in the evaluation of our method with the MoleculeNet benchmark datasets, accurate prediction performance were achieved. And apart from that, it is important to note that, of course, our method solves one of the problems that can be taken into consideration, but there are a lot of other tools available for drug discovery, especially deep learning aided drug discovery, and many of them are publicly available. Thank you.